that the cells are getting too crowded and we want to give them more space to grow. So we'll take them from one plate, lift them off using trypsin, which is a protease, so it's going to chew away the bonds from between the cells and the surface, and lift them up, and then we are going to put them into two different plates, so they have larger surface area to grow with, and they can continue to divide. They are going to be in these incubators, right? These are cell culture incubators. They are water jacketed, CO2 attached, so they can maintain the right environment, similar to how we have inside our body. They are set at 37 degrees Celsius and 5% CO2 concentration. You're going to take one of the plates from the bottom shelf. You don't want to leave the incubator door open very long because you don't want it to change temperature or change the environment, right? Once you have your cells, you're going to walk over to the microscope. These are the inverted microscopes. The cells are attached to the bottom surface of the plate where the ECM-like material is. So, and then there is media on top. So we cannot use the normal microscopes, the compound microscopes, since they have the objective lenses on the top. With the inverted microscopes, the objective lenses are at the bottom. So we can look at the cells underneath that are attached to the surface. So this particular microscope is connected to the computer, so you can look at the cells here, or you can look at it in the eyepiece. So you want to look at them, and you want to decide how confluent or how much the cells are, how many cells are there. So here are my cells. You can also look at them on the screen up there. As you can see, majority of the surface is covered in these cells, right? They are flat. You can see the nuclei inside them. This is just a bright field picture, so the contrast is not there as much. But you can clearly, and then you see some floaters. The floaters are cells that are probably dying or dead.
Is that the ones that are kind of stacked on top?
Well, maybe they were trying to become stacked, but they have not. These cells are not going to be doing that, so most of them are going to be dying cells. Some of those cells that are still attached but rounded up are cells that are dividing. So you will look at that. This one is about 85-90% confluent. There is very little space inside that is still left to be covered. So definitely this one needs a new home. And so that's what we will do now. So once we look at it, we look at the cells to make sure they're healthy, they are clean, right? They don't have any infection. And we always write down their confluency. So this one is 85-90%, that's what we write down. And then you bring it into the aseptic space. So now I'm ready to work with them. The first thing you're going to do is remove the media that's already on top of these cells. So you're going to take one of your pipettes and work with them the same way, except that you don't want to touch the pipette anywhere, including the tip, especially the tip. So you take it off. You lift the top of your plate just enough to get in. You do not want to expose the cells too much to the air, right? Because that's going to contaminate them. Also, you always want to hold them tilted so they don't dry off. And then you will get rid of this liquid in the liquid waste, right? And you see the change in the color of the media because you have conflict in there, and that's changing the pH. And the DMEM that we have has phenol in there that changes color with the pH. So it has the phenol red, that's what gives it the red color. Now we put it back in here like this, take it out, and this is now our waste. We are not going to use it. Next, we are going to wash our cells with the PBS. What this will do is this will remove any other waste that's still on them, and it will also remove traces of the FPS, the media, from there. The serum in the media is very sticky, so those proteins tend to get stuck to the cells, and they make it harder for trypsin to work because it's a protease. It needs to be able to see the amino acids to cleave them. So again, you are going to take a new pipette, don't touch the tip, work close to the flame, get your PBS. You just need enough to cover the plate, you don't need exact volume. Always work close to the plate, the flame, and also don't squirt the media or the PBS on top of them, right? You want to go along the edge of the plate so that the cells don't get lifted off and disturbed by liquid squirting on top, right? So you tilt it and you work along the side of the plate without touching the tip to the plate itself. As long as you don't touch it, I can reuse it to remove this wash as well. So I'm going to just rinse the cells by gently either rocking them back and forth like this, or just doing this, swirling it, or just, go along like this, and that's all it needs. And then you're going to remove the wash buffer. If you're unsure if your pipette is clean, by all means get a new one. Work close to the flame again. And now again, just lifting slightly, just enough to get in. You want to lift out the liquid. You want to remove as much of the PBS as you can because you don't want to dilute the enzyme, right? The trypsin. Take it off. And for the trypsin, it's already pre-measured for you, so don't worry about using any pipettes. Just lift it up, dump it, and that's all you're doing. Dump, and then swirl to make sure the whole surface is covered. This is waste, and now you let it sit. Here, each trypsin is different, because maybe it's been out too long, maybe it's been freeze-thawed multiple times. So they'll usually start out really high efficiency. Over time, they lose their effectiveness as much. Also, the cells, different cells have different efficiency to it. So some cells trypsinize within a minute or two. Some cells take 5-7 minutes. So you have to keep an eye on your cells by looking at them under the microscope every 2 minutes to watch when they are lifted off. You don't want to over trypsinize them, so you don't want to get to the point where all of them are lifted, because at that point some of them are getting damaged and will never make it. They will die because their cell membranes are disrupted too much by too much protease activity. On the other hand, you don't want to under trypsinize it, because if you under trypsinize, majority of the cells will remain here and they will never be lifted off, so you wouldn't be doing the job correctly, right? So you'll be losing most of the cells to the original group. Okay. While we are waiting for our trypsin to work, you're going to get your new plates ready. You're going to have one plate per person in your group. So you're working in pairs, so each person will get one plate. You're dividing them into two, right? So you want to write your first initial and your last name, so I know whose plates they are. You want to write the name of the cells that you are splitting. These are MDA 468 cells. These are breast cancer cells. And you want to write how many times it's been passaged. These have been passaged 30 times. So today, when you split them or passage them, it will be passage 31, right? Because you are splitting them another time. And then you want to write the date. So today is what? 21st. So the second person will do the same thing. They will write their name. They will write the name of the cells. They will write the passage number, passage 31, and today's date. The second thing you're going to do is that now you're going to go ahead and add 8 ml of media to each one of these plates. So they're ready to go and receive new cells. This allows to have some liquid in there so the new cells get more evenly distributed when you add them on top. And you need this one close to the flame because we are going to make the cell suspension in that empty tube. So I'm going to use my media. I'm putting it close to the flame. Get a new pipette. Lift up slightly to get your 8 ml. If you go all the way up to the double lines, that's 8 ml right there. You're going to go ahead and put it in your first plate. Right now it doesn't matter if you put it in the middle because there are no cells there, but you don't want bubbles, so be gentle. Get media for the second one. You can come to the other side. And add that, lifting it just slightly. Do not lift it too much. And that's it. And now you are ready to receive cells in those new plates as soon as they're trypsinized. Also,
can you start again?
Yeah.
Sorry.
So if you look at the cells, they're rounding up. They're not as flat anymore, right? And they're lifting up. You see cell clusters floating away. If I were to shake it like this, and this is just, maybe like another 30 seconds, 1 minute, no more than that, and this is done. So you want to stop them when they're about, 60%, 70% lifted, the rest are attached. So right now it's about 50-50. But you see how they're pretty much getting ready to lift off. We're going to create what we call a cell suspension. And a cell suspension is simply a mixture of cells with the trypsin and the media to divide amongst our new culture plates, okay? The FPS in the media, the sticky proteins, inactivate the trypsin. That's why we use the media to add to the trypsin right away to stop the reaction, to stop the trypsinization process. So I am going to get about 4 ml of my FPS containing media. I'm going to now squirt it on top of this plate, and as you do it, you will see the liquid becoming a little bit more opaque and the plate surface becoming clear. And that's because now basically the cells are
Part of the solution.
yeah, they are getting in the solution, and you are not going to have them on the surface, right? They're lifting off from the surface of the plate. You pipette them up and down a few times to make sure you collect all the cells from the entire surface, and also so they become individual, like single cell suspension. You don't want them as clumps, right? Because if they are in clumps, they will not work very well. So that's that. I feel like this is leaking somewhere.
It smells like
Okay, well, I'm almost done. Okay, so now we are ready to divide it amongst the two plates. For this one, what we are going to do is each person will do their plate, okay?
Mm-hmm.
So we will have the first person take a pipette tip, a serological pipette. We will go in. I would again squirt it up and down once, make sure it's well mixed. Then you make sure all of it is out. Then take just 1 ml of the cell suspension, okay? And that is what you're going to put in your plate, okay?
Mm-hmm.
As soon as you do that, close the lid and swirl it to mix the cells so that they are equally, pressed all over there, equally spread. The second person will do the same thing. They will go ahead, take their pipette, a brand new one, pipette the cells up and down, and get 1 ml of cells, and then put them in the second plate. And then they are going to go ahead and take this out. And then swirl them. So these are your plates that you are going to be tested on. That's what we are going to grade for your aseptic technique. With the remaining liquid that's there, what you're going to do now is you're going to put 8 ml of this liquid of the DMEM, the fresh DMEM, back into the original plate. So that anything that's left behind can be recultured. You don't want to waste good cells, right?
Mm-hmm.
You are going to take your cell suspension, take all of it out. You're going to put a small aliquot of it in this yellow tube to count, and the remaining back in the original plate. And you're done. Now this one doesn't need to be sterile. This is done. All these cells need to go back into the incubator immediately. Okay, so once you start with your cells, you're kind of on a timer. They don't like to be at room temperature. You want to go through the steps quickly and be done with it within 10 to 15 minutes.
Do you want to also
So once you have returned the plates to the incubator, you want to get rid of all the stuff that is a biohazardous waste. The serological pipettes will go into the biohazard bin that says serological pipettes only. The liquid waste, once contaminated, decontaminated, can go down the drain, and the rest of the supplies all go into biohazard bin for the remaining stuff.